Good evening. It's a pleasure to have with us today Professor Nathalie Balaban from the Rakach Institute. Nathalie did her undergraduate and PhD, undergraduate in physics here in Jerusalem, and PhD in biophysics, more or less, in the Weizmann Institute. Since then, became a real, what she calls, biological physicist in Princeton, or mainly in Princeton, and is really considered one of our stars in this field. She has really a very impressive list of publications in the most prestigious places, and she is a central figure both at the Sudarsky, the biological computational biology center, and I think also the nanoscience. What I could find, but anyway, she is interested really in this exciting connection between genotype, phenotype, and fitness in evolution in general. And she's doing both theory and experiments in this area, and I'm sure it will be interesting.
Thank you, Nathalie, for the introduction. Yeah, indeed, my lab is located in the physics department, and we work on biological systems, and it's something that is gaining more and more attention recently, because not so much because of physics, but I think because the biology has become so well defined in latest years, and you have so many amazing tools in order really to control the biological system, that you can start thinking that you may gain understanding of biological system as we are used to understand physical systems. So in my lab, of course, from this very general framework, we focus on one phenomenon at the conceptual level, which is the fact that in biological systems you can see a lot of stochastic behavior. And understanding stochasticity, understanding the source of stochasticity in biological system is something that makes you ignore a little bit the details, roughly, and then see what is the mechanism at the conceptual level that controls this stochasticity. So this is the molecular level. But stochasticity at the molecular level has a phenotype at the microscopic level of the organism or the cell, and of course it's very interesting to see the relation between the two, and eventually to understand how stochasticity has an impact on the evolutionary process that occurs in this organism. So just to focus the frame even more, I talk about non-genetic variability, namely the fact that variability that I'm talking about is non-genetic, so it's not coded in the DNA in principle, and focus on the variability of growth. So growth is a major parameter, especially at the level of bacteria. How bacteria grow is a crucial parameter in their behavior in the wild and definitely on evolutionary time scale. So this is the parameter that I'm going to focus on and show how variability of growth actually it can be beneficial. The lab is mostly experimental, so we develop tools in order to study variability quantitatively, and we see at the end how a molecular module really can control variability at the synthetic level. So that's the overall frame. How much time do I have? I didn't ask this question.
About an hour.
About an hour? Okay.
Well, it's a bit late now.
Okay. Okay, so variability in biological systems, typically we are taught that when you have one phenotype times one environment, you expect to see just one phenotype. Right? So these mice are clonal and are grown exactly in the same condition. You expect them to look very similar. Clearly, you can get two phenotypes if you grow these mice in a different environment. Okay, so this is gene regulation. This is well known, and this dogma works quite well. Although even there, there are some new aspects, but at the level of the organism, the whole organism that contains billions of cells, it works not too bad. But when you look at single cells, and in the lab we work with bacteria, a little bit with yeast, and now with lymphocytes, at the level of single cell you see, yes, thank you see quite a lot of variability. Here these cell express fluorescent proteins, and you can see that whatever you do, there is some variability in the phenotype that you observe at the single cell level. So, in order to really see that this phenotype is some non-trivial variability and not just, if you expect almost every parameter to behave as some distribution, as a Gaussian distribution or log normal distribution, of course you expect some of the cells to be in the tail of their distribution. But what we are looking for is the non-trivial variability that really shows you distinct phenotype within the same environment. And one such phenotype that was actually hinted many, many years ago was the behavior of bacteria when they are exposed to antibiotics. So this is, of course, one aspect where variability is interesting even for hardcore microbiologists. When you put bacteria at time equals 0 in antibiotics, this is the number of surviving bacteria. Right? So what you see, and this is what antibiotics are supposed to do, is that they kill your population very rapidly. Right? But after a while, if you do the experiment on a long time scale, you can see that some of the population is killed at a much, much lower rate. Okay, so this is the first hint that you don't have a uniform population, because a uniform population will be typically described just by one time scale, either this fast time scale of minutes or this time scale of a day, about a day. So, of course, when you do a bulk experiment, here the experiment is done very classically. You have a tube with many, many bacteria, and each time point you place the bacteria and count how many surviving cells you have. Okay? So you don't really look inside this tube in order to see that the conditions are identical for all bacteria. And people every time they saw something like that at the beginning, people typically thought that maybe there are aggregates that break, the condition into cells that are in aggregate and cells that are not in aggregate, or things like that can define you very trivially to different populations. So in order to really study viability, you want to control the environment. In order to be sure that all your cells are put in the same environment, you want to control it. And what we do is that we fabricate chips that are made with microlithography, so it's the same technique as for making computer chips. Basically, it allows you to pattern in space small dimensions. And we make, for example, grooves in which we trap single bacteria, and on top of these grooves, we put a membrane in which we can flow conditions. And here, what the bacteria see are constant conditions that you draw them on. Okay, you can also vary the conditions if you want, but at least you completely control the environment, and you can believe that all these single cells that you look at under the microscope are in the same environment. So here, this is how it looks like when you look at it roughly. Bacteria that grow in these grooves make linear microcolonies, right? And you can measure the growth rate. And now you can do the same experiment that actually Bldg did, microbiologist back in the 40s, and subject them at t equals 0 to antibiotics. So first, we grow them. So when they grow, they go on these grooves. Each point here is a bacterium. Hopefully this is clear. Each small rod is a different bacterium. And the population is clonal, so they have the same DNA, and you can subject them to antibiotics. This is what antibiotics do, right? They clear off your infection. But if you wash the antibiotic and wait long enough time, you can see that some of them manage to survive, and then now they grow. Okay? So just to make sure that this viability, the fact that some bacteria did not grow is non-genetic, we can subject these bacteria now to the same antibiotic, second round, and you can see that they die at exactly the same rate as the original population. So it's not a genetic effect, it's not resistance. You've probably heard about resistance. It's not resistance. Resistance occurs at a much, much lower rate. This is persistence, and it occurs whenever you put any bacteria with any antibiotic. So when you look at these bacteria that survive, you can look at them more carefully. Actually, yeah. You can look at them more carefully and look at these surviving bacteria and try to understand, are they just at the tail of the distribution, or are they inherently different as the bacteria that die immediately? And here you can see these bacteria are going to survive the antibiotic treatment. All the other bacteria die. You can look at them here. And when we wash away the antibiotics, they are the only one that managed to survive in this frame. So, in fact, if you look here, in this part of the movie, there was no antibiotic yet. All the bacteria, these bacteria grew very nicely, right? And you can see that the bacteria that are going at the end of the movie to survive are the ones that hardly grow. And in fact, all this riddle of why bacteria survive antibiotic treatment can be mapped to the fact that there is a growth rate variability in bacterial population. And the fact that bacteria do not grow makes them much more persistent to antibiotics, because antibiotics typically target actively growing cells. And it's a very similar mechanism if you take cancer cells. Anti-cancer drugs are going to kill actively growing cancer cells. If you have a cancer cell that for some reason doesn't grow, it will not be killed by the anti-cancer treatment. So this type of variability, of course, can be very important clinically. What we want is really to understand the dynamics of this process. We have two populations, and now we really understand their physiology, so we have markers that are differentially expressed in these two subpopulations. So the population of cells that do not grow, the persister cell, or the ones that are prone to be persistent, are here expressing a red fluorescent protein, and when they grow, they start expressing a green fluorescent protein. So growth is easily visualized here just by green. And when we put antibiotics, you can see that only some of the red ones have survived. It's a stochastic process. So the probability to die for a non-growing cell is much lower, but it's non-zero. So only the red cells have survived, and they have, once you take away the antibiotic, they have a probability to switch back to the growing phenotype, and then they will turn on green. But again, both the green and the red bacteria here come from the same clonal population. So once you understand really the system and you can mark each population with a different marker, it's very clear that you are in the presence of two populations. And these two populations actually are what underlies this killing curve, where with one very fast rate, this is the killing rate of the green cells, and this is the killing rate of the red cells. But now if you want to do the experiment without antibiotics at all and just characterize the growth rate variability of population of bacteria, and you want to see a distribution of growth, so it's not like taking the cells to a spot and having very high statistics because you measure some expression levels. You want to measure many thousands and thousands of bacteria growing under the microscope. And it is not an easy task. You can do it the classical way, just by plating cells. Here you can see bacteria, E. coli, growing under the microscope. And you can see some cells start growing immediately and others are arrested. They can stay here for a day and afterwards start to grow at a much later time than the typical population, and in typical assay you just won't see them because they will be completely overwhelmed by the fast-growing population. But we want to get a very precise distribution of this exit time, exit from dormancy. And see, first we're interested, but I'm not going to talk about it, the mathematical shape of this distribution, but in order to really characterize two phenotypes, we want to see something that is close bimodal distribution. And at the end of the day, we can see that in this growth phenotype that we define, we can measure, and here this is appearance time, so when this is the growth time, so when do they exit this dormant state, we can indeed measure it, and this is the measurement that I'm going to describe in a minute, that really defines two growth phenotypes, some that grow at early times and some that grow at times that are longer by more than an order of magnitude. Okay? Didn't say it at the beginning, but please do interrupt me. I've no. Okay.
So which antibiotic is it, and is it common to different types? Because each of them targets a different.
Right. So that's, of course, a very, very good question. Clearly, in antibiotics, you have antibiotics that are completely different mechanisms. As long as the antibiotic kills more efficiently actively growing cells, and most antibiotics are like that, this phenomenon is going to be very important. So for what I showed is ampicillin, so it's beta-lactam. It kills bacteria when they try to form new membranes, but you get exactly the same thing if you target for DNA replication, for example, which is a completely different class of antibiotics. Other questions?
What is the real-time scale of the movie? What's the real rate?
So the movie itself is about a day and a half. Oh, this one I think is several days, but the microfluidics was a day and a half. Okay, so does it make sense? I mean, this is E. coli, right? E. coli is a pathogen, or at least it grows inside us, and we know that it's a very fast-growing bacteria. It divides typically every 20 minutes. So how can some bacteria choose, in a sense, not to grow? Does it make sense, or is it just a byproduct of some deficient cell cycle process? So to really discriminate whether it's a byproduct or not is a very tough task. But at least conceptually, you can ask, when is the fact that some bacteria do not grow provides an advantage? Okay. Now, as I told you, if you have one phenotype and one environment, and this phenotype stays constant all the time, you expect just one phenotype to be optimized for that. But if now you still put all your population in one environment, but this environment fluctuates between good and bad conditions, then the situation is not that clear. So, for example, if you have only the green bacteria, only fast-growing bacteria, and you put an antibiotic, not only antibiotic, but many different stresses, will just wipe off all growing bacteria. And we've tried other stresses that I'm going to talk about, for example, phages, which is viruses of bacteria. So typically, those stresses are going to just wipe off this growing population. Now, if stochastically this population generates some bacteria that do not grow, these bacteria are the ones that are going to survive. They are going to stay in a non-growing state for some period of time, a typical time scale, but then they are going to regenerate the whole population again. Okay? So it's like a bet-hedging strategy, or not putting all your eggs in the same basket. Right? So it's very appealing to think that the variability that we measure actually has evolved to sustain such environments. And at the theoretical level, it can be shown relatively easily, and it's very simple conceptually, is that if you have environments that are bad and good with typical time scales, the optimized population will be growing and not growing with the same time scale. Okay? So in principle, just by measuring the time scales of growing versus non-growing of your bacterial population, you can infer which environment is optimized. Now, the implication of growth rate variability is, of course, at the evolutionary level, it's very interesting to study it, and we are performing evolutionary experiments in the lab. So we really take bacteria and subject them to cyclic environments and see whether we can see this relationship between environmental fluctuation and growth rate. But this is a long-term experiment. As Matal notes here, experiments takes time. And but at the clinical level, of course, these non-growing bacteria that are protected from many antibiotics are a big problem. Now, we usually don't hear about them. We hear about the resistance because in normal hosts and for most of the diseases, these dormant bacteria are just wiped off by our immune system. But now, if your immune system is deficient, as in cystic fibrosis or in HIV, or if the bug that infects you is perfectly adapted to your immune system, like in tuberculosis, then even a single bacterium that survives can restart a whole infection. Therefore, it's really important to understand what actually can control their viability. So we were, for several years, we were searching for the molecular mechanism responsible for growth viability. So this molecular mechanism, despite the fact that to grow, not to grow, is not a genetic effect, I mean, it's not a genetic difference, to have this viability of growth has to be encoded somewhere in the DNA. And when I say we're looking for the molecular mechanism, we're not looking for the molecular mechanism that arrests growth for the specific protein, but we're looking for some noise amplification circuit that would take stochastic fluctuations that always exist in cells and amplify them in order to get two different populations. Right? If you don't have such a mechanism, you would expect a log normal or Gaussian distribution, some well-behaved behavior. But what we see here is some bimodal distribution, so some noise amplification has to take place. One well-known example of noise amplification, if you have a gene A whose level of expression controls the production of gene B, and vice versa, so this is a positive feedback loop. Typically, the lactose system is controlled by this type of topology. So when you have a positive feedback loop like that, and you solve the equation, the rate equations that are very simple, just the rate of expression of A is controlling the rate of expression of B and vice versa, you get two stable points. So if you solve for the steady state, you get two solutions that are different. And these two solutions already can generate the viability, so small fluctuation in the initial condition will turn the population into those that reach state 1 and those that reach state 2. And in the lactose system, this was observed. So we were looking for this type of noise amplifiers. Now, in order to do that, we really need to be able, we're going to dissect the genetic circuit, we need to be able to measure, I told you, many cells, thousands of cells, and measure their growth characteristic of each one. So we can do it in the microfluidic device, but these are very long experiments that take the microscope for days sometimes. And we wanted a much simpler method to measure how long does it take for a bacterium to reach a rich medium and start growing. And for that, we went back 200 years ago, actually, to the days of Koch and Petri, and just plated bacteria on Petri dishes. Okay? So if you plate bacteria on Petri dish, on a Petri dish, I hope every one of you has done it at least once, maybe. So if you plate bacteria on the Petri dish, typically you plate it, you go home, you come back the day after, and you see colonies. Now, if we have bacteria that grow very differently, we expect their colony to appear at a much later time. Right? So just the appearance of colonies is a proxy for measuring actually the exit time from this dormant state. So we took office scanners, and on top of them we put the Petri dish, because we are not just going to look at them for a week, right? So we put them on scanners, and the computer is hooked up to the scanners and controls them completely. And this is the movies that you can see for each of the Petri dishes. So you start with no colonies, and each time a colony appears, the computer program detects it and says, here is a colony. Okay? So this is the work of Eric Levin, that's with by Ofer Friedman. And this is how it looks in real space. It's much less neat than PowerPoint schemes, but and this is how the movie looks. So here we have two conditions. Never mind which condition. Cells that exit dormancy very quickly and cells that are spread. And as colonies appear, we can detect them and plot here the number of colonies that have not appeared yet. So this is one time constant, and here you can see actually a continuum of time constants in some conditions. And you can see that some colonies really appear at much, much later time in one of the plates. So the computer program actually really follows the growth of each colony. And this is the area of each colony and the time it was detected. And from that, this is the way we construct histograms of growth for thousands and thousands of cells. And there you can see, for example, the bimodality. You can make also a two-dimensional plot of this exit from dormancy, where here is the appearance time, so when each colony was detected, and here is how fast it grows in area. And you can see that the colony that was detected a day later still grow at the same rate as the one that were detected at early time. So it's not a difference in the growth of the colony, just a difference of when did they start growing. And O'Neill in the lab really did a screen of many different types of bacteria, and each of them have different growth characteristics. So these growth characteristics are very interesting to study, but the main question here is, how do you really generate this variability in a uniform population, and what controls the typical time scale of the growth arrest? So any questions up to here? No. Okay, so where do we start? We have a phenotype, and we don't really have a clue of what controls the phenotype. So as usual in genetics, you start from the mutant. So there were the mutants isolated. With a mutation, nothing is known about this mutation except the fact that if you have it, if the bacteria has it, it has a much higher probability of being in a dormant state. So still the population is bimodal, but now the amount of growth arrest cells typically in the population is much higher than for the wild type. The mutation is in the toxin-antitoxin system. It's in the toxin of the toxin-antitoxin system. And when you do the histogram with the scanner system of the wild type and the mutant, you see that indeed in the mutant most of the population is still as in the wild type, but instead of the very small population of dormant cells that is in the wild type, here the population is at much elevated level. So this is the starting point. Okay, we don't know exactly where growth variability is, but we can control it by just having one point mutation. And we can see that the mutant has two time scales in the population, the time scale of the wild type here, and another time scale that is at much longer time. So the subpopulation of the wild type is just much smaller, so we don't see it here, but it will also eventually change. What differs from the wild type and the mutant is just the level of this persistent bacteria. So what is a toxin-antitoxin system? I suppose you haven't heard about a toxin-antitoxin system, right? So what is nice about this module, especially for physicists, that it has just two genes, okay? No big developmental pathway, just two genes. One is toxic to the cell, arrested growth or even kills it, and the other one, its only function known so far is that it cancels the effect of the toxin. So the toxin is the antitoxin. Now, why on hell would bacteria generate something that kills them and something that prevents their killing? Not very clear, but these toxin-antitoxin systems were discovered first on plasmids. So plasmids are small DNA elements that are in cells, but bacteria can get rid of them, right? Now, if a bacteria is infected with a plasmid that contains this toxin antitoxin module, it will produce the toxin and the antitoxin. Now, if it gets rid of the plasmid, then the toxin and antitoxin will stay in the cell. The antitoxin has a very short lifetime, so it will be eliminated, and now there is no production of antitoxin anymore, so this cell will die. Only cells that are generous enough not to throw out this plasmid but replicate it will continue to live. Okay, so they were called addition module because once you have the plasmid, you cannot get rid of them. So there it made sense. But then it was discovered, actually it was discovered at the Hebrew University by Gadi Glaser and Hanan Kulka. And the first module that they saw that, they called it maze. But you heard Hannah because nobody understood what were they doing on the chromosome. They're supposed to maintain plasmid. Chromosomes do not need this maintenance system. So it was not clear, but since then there were many discoveries. They exist in all bacteria, okay, at certain levels. In tuberculosis, now there are 80 such modules from the chromosome. And since the question maze, many work has been done, and lately there are 9 possible functions of toxin antitoxin systems, which means that we don't have a proof. So when we bumped into this module that actually has some control of the variability in growth rate, we were looking both to try and say something new about this toxin antitoxin system, maybe understand them better, and understand how come they make one cell grow fast and the same genetic network will allow other cells to stop growing. So,
is it the only one in E. coli?
What do you mean?
The toxin antitoxin.
No, in E. coli there are now about 15.
And this mutation is in one out of those 15?
Yes, just in one toxin.
One toxin, one
Yes.
unit.
Yes. But we study other things and they all behave the same. So the mechanism is quite general. So what you can do in E. coli is that you can completely take apart modules, right, and control them separately. So this is what we did. Take off the module from the chromosome, put the toxin under an inducible promoter that you control, put the antitoxin on another inducible promoter, and do the games of expressing one and expressing the other separately. So here the level of the antitoxin is constant, is set, and we change here the level of expression of the toxin. So what do you expect? You expect that more arrest will occur. The toxin arrests the cells. We showed that at physiological level it arrests the cell, it doesn't kill them. And if you plot this distribution as 1 minus the CDF, you clearly see the different time constants. This is without the toxin or with small level of the toxin. At high levels, the time scale goes up and up. And clearly the toxin level controls the growth arrest duration. Both the growth arrest duration is mean and also its variance. So you can see that the variance here is huge. So this was one answer, what controls growth arrest duration. At least in this system, it's clearly the level of the toxin. But what is interesting to note is that at its intermediate level, and here we are talking about the wild type toxin, right? We're not talking about the mutant yet. At intermediate level of wild type toxin expression, you can see a coexistence of 2 time scales. Here this is about 15 minutes, and this is already a few hours. So you can, with the wild type toxin, you can reproduce this phenotype at the level of the population of having both growth arrested here in the tail and not growth arrested cells. So, but of course, doing it at the level of a population with plasmid has its experimental limitations. So you have to do it at the end in single cell under the microscope. And now we wanted really for each cell to measure both how much toxin it expresses and how long it is arrested. And this is a threshold here is how much toxin is expressed, and here is how long it took for the bacteria to start growing again. And you can see that for quite high level of expression of the toxin, we don't see any effect, which was at first a bit surprising. But then, as soon as you reach a threshold, then you see a very big effect, and very strongly. So basically, this curve already explains to us why do we have two types of cells in the same population. You have a threshold. Now around this threshold you have some background noise, right? And now the cells that are above the threshold will be growth arrested. The cells that are below the threshold will just grow normally. And actually the model of the toxin-antitoxin system, the equation that governed the behavior of this very simple module actually show it very clearly. This is a work done by A. Longer from the group of Safra Campus in physics. He made a stochastic simulation of this equation, but you can see already in the mean behavior that you have a threshold mechanism. And why? What is this threshold mechanism? Basically, it's very simple. The cell produces the toxin. The cell produces antitoxin. It produces usually more antitoxin than toxin. So as long as you produce more antitoxin, and the interaction between the toxin and the antitoxin is very rapid and effective, then each new toxin that is made is going to be destroyed by the antitoxin. Okay, and so there will be no effect. Now, as soon as the rates of expression of the two are close to each other, you have what is called an ultrasensitive process, but it just means that now every small deflection, every noise in each of the levels, and you'll get many cells that are growth arrested. So, actually, it's because in a difference of large numbers, and this gives you a very trivial mechanism for amplifying noise. All you need here is a very strong interaction between the A and the B. Okay? So the model tells us indeed that there is a threshold mechanism. So here, as function of the expression level of the toxin, more and more cells are going to be growth arrested, and what determines the position of the threshold is the level of the antitoxin. So if you now, before I showed you that we control the level of the toxin. Now, if we control the level of the antitoxin, we expect to see that, and not surprisingly, this is exactly what we see when we change the level of the antitoxin in each different experiment and vary the level of the toxin. Now, so when you have a model, and the model is simple enough, then it's appealing to try and get some more out of the model than just reproducing experimental results. So we still did not have an answer of why is our mutant so much so much more persistent cells, so much more dormant cells. What we know about the mutant is that it expresses more the module itself. So the module itself produces the toxin, produces the antitoxin, they bind to each other, and then they form a repressor that represses their own expression. So it's a negatively regulated module. Okay? Now, what we knew is that in the wild type, this repression, the strength of this repression is much lower than in the wild type. So if you take off completely the module itself, then this promoter is very active, no repression. So this is no repression, intermediate repression, and strong repression. So this module itself is more expressed in the mutant. Okay, this we can measure just by measure the transcription to GFP. So this we know that this arrow is weakened. Now if we ask the model, okay, the input is that this arrow is weakened. What is the parameter that is affected by the mutation? Is it the binding of the toxin to the antitoxin? This is one parameter that can be affected by a POG mutation in the toxin. Or is it the repression itself? So the binding is not affected by the repression, is less active, and therefore we have more of this module here. could be either parameters, but if you look in the model, what? Can be both. Right, right. Definitely. But, you know. So, if we ask the question, which is the more probable to be responsible for the effect, then we have a clear answer. Of course, it can be a combination of the two. Probably is. So, here is the very simple paper. So, this is the production of the toxin, the production of the antitoxin, and the color tells you how much of the population is in the growth arrested state. Okay? So before the experiments were done around this axis. So you go from no growth arrested state, here is the threshold, and then here all your population is growth arrested. Okay? Now if you look at the wild type is located here. It is far away from the threshold. It has only a very small number of persistent cells, of dormant cells. Okay? This is and it produces more antitoxin than toxin. Only once in a while you will see a dormant cell here. Now we know that our mutant is somewhere here. It's about 50-50 dormancy and non-dormancy. So the mutant has to be located in this transition region. Now if you alter this parameter, the binding to DNA, the binding to the promoter, you are going to move around this line. So yes, you don't bind here, so you produce both more B and more A, right? They are co-expressed, the B and the A. So you're going to move around this line, far away from the transition region. So this is unlikely to be the parameter that is affected. Now, if you decrease the binding, I told you the crucial parameter here for this amplification of noise is a strong binding of A to B. So if you decrease the binding, you're going to change the whole phage space, and now you are going to move from here to here, so you are going to produce more of both, but the whole phage space changes, and now you may be in the transition region. So it's more likely that the binding between the two is what is going to be affected. So then we wanted to measure that. So in order to measure that, we want to measure binding of two proteins in single cells. So we do a translation fusion to the toxin and to the antitoxin for the wild type protein here and for the mutant protein here. So this is B, this is the antitoxin, okay, and this is the wild type toxin and the mutant antitoxin. Already here, you can see that B and A, when it went at the co-localized, and here they co-localize much less. So it's already a hint that the interaction here is decreased. But of course, in order to really measure it quantitatively, we use threat. Everybody knows what threat is? No? Okay, so threat is a very nice technique to measure binding of proteins here, for example, which relies on the fact that when you have two molecules that are fluorescent, and you excite one of them, when you excite a fluorescent molecule with light, it will emit a different light, right? So here we have a green fluorophore, we excite it with blue light, and it will emit a green light, right? Now, if you have a fluorophore that is red and can be excited by the green light, instead of emitting the green light, you are going to get red light out of it. So if you measure, and it occurs only when the two fluorophores, it's a quantum mechanical effect. It's you can see it as the fact that the virtual photon from here is the green photon, instead of being emitted from there, excites the other fluorophore, and therefore it can occur only when they are very close to each other, nanometers away. So it's not going to happen for molecules that are freely diffusing in space. It's only going to occur for molecules that are really tightly bound. Okay? So if you excite the green fluorophore with blue light, and the red fluorophore is far away, further than this few nanometers, you're going to get a signal. Let's call it 100%. Now, if the red fluorophore gets close, the signal is going to go down, the signal of the red is going to go down because some of the photons, instead of going to the green path, go to the red path. And if you take them away again, the signal is going to go up. So it's a very standard technique for measuring interaction in bulk. In single cells, it's a little bit more tricky. And for that, what we do, so we have two proteins, N and B, they bind together. They each have a fluorescent protein in their tail. So the fluorescent proteins are close by too. We excite the green light, and we get, when they are close, we get only 95%, around 95% of the signal. Now we are going to get rid of the red fluorophore by bleaching, and now the signal is supposed to go up. So this is, this is a very clean signature because you bleach, so you excite this fluorophore with a lot of light, and instead of seeing your signal go down, you have to see it go up. So this is what we did. We measured several times the green fluorescence, so this is the 95% in principle, and then we bleach, we take away the red fluorophore, and you see that for the wild type protein the signal indeed goes up. And it doesn't go up, or hardly goes up for the mutant protein, and it goes even slightly down for the control with free diffusing red fluorescence. So this tells us that indeed, most likely the parameter that is affected by the mutation is the binding of the toxin to its antitoxin. So in those modules, a very slight change in the parameters of the toxin and antitoxin really control the variability of growth in the population. And it's quite general. So if you do similar experiments with other toxin and antitoxin systems, you get the same effect of this variability of growth. So in principle, these modules are ideally suited to control growth variability. Yeah?
So this is all in the model, but what happens in the wild type population? Do you also have a differential expression of the toxin antitoxin or differential binding?
Okay, so that's a good question. What you need to measure in order to see the effect is not the expression level of the toxin. And you really need to know how many free toxin you have. And this we didn't do, okay? You can mark the toxin with a fluorophore, but knowing exactly how many of them are free is much more difficult. So we think of how to do it. But what is clear is that the wild type persistence is governed by the same dynamics as the mutant persistence. So this is one hit. But yes, when are these modules coming into play is not clear, right? What we can say is that any environmental condition, here it was done by mutation, right? But you can have environmental conditions that will affect the binding of these two proteins. So environmental conditions that will affect the binding of these two proteins can move the wild type from having low persistence to having more. Okay. Much time with 10 minutes. Everybody agrees? Okay. So this is one thing that we could say about these dormant bacteria, but in order really to understand their physiology more, we wanted to have some more quantitative measurements and to grow, not to grow, because they're really growth arrested, this type of persistence. So always what we can say when we look under the microscope is that they don't grow. So we really wanted to measure a response function of these persisters in order to understand when is a switch from growth to not growth occurring in real time. So what is a response function? It's actually it's a very gross phenotype. It's just measuring the ability of the bacteria to respond to an external signal. So for that reason, we have an artificial system in the bacteria. We have a red fluorophore controlled by an expression system that is controlled by a chemical signal from the outside. So when you grow bacteria with the chemical from the outside, they turn on red. Okay, and they are green all the time because they're in the chromosome a green mark. So you can ask whether persistent bacteria that are dormant are going to respond at all to the external signal. In principle, if they are dormant, they're supposed to be completely shut down. But then looking at this expression system quantitatively can give us a hint of when does it happen. So for that, we developed a software to really follow and track bacteria as they divide and measure the fluorescence at the same time. And then for each bacterium like that, we have an induction curve, a response function. So at t equals 0, we have the signal, and then these are traces of each of these bacteria. So you want to understand for the function of the form, why does it look like that? And this is quite simple. It is maybe one of the only examples where we can understand the functional form of gene expression. Because you have no feedback, you just have a very artificial system that you control. So in principle, the artificial system is at the level of the DNA, some plasmid, and you have, you can say that there is a rate of expression from this DNA of a protein. Okay. Now this rate of expression is controlled by your external chemical signal S, which controls this arrow. More signal, bigger arrow, and the protein can in principle degrade at some rate. So this very schematic view of gene expression can be modeled just by one rate equation, and you expect, if your signal is a step function, you expect the protein level to go like 1 minus an exponential decay. Okay? This is what you would expect from this very simple signal. In fact, if you neglect the RNA level, it doesn't work. So you need to take into account RNA level. And this adds one more equation. But still, you can predict that the RNA level will look like that, and the protein level response function will have this shape. Okay. Now, this shape is controlled so we can fit it, right? But we don't really look too carefully at exactly which fit is the best. What we try from each fit for each bacterium like that is a typical time scale for reaching steady state and the level of the steady state. Right? These are the main two parameters that will characterize the response function of each cell separately. So now back to our dormant cell. A normal cell has such a curve. What do we expect from a persistent cell? As I told you, there are two parameters: the amplitude of the response and the typical time scale to reach the response. So dormant bacteria must be just much slower. So they do exactly this. They have the same response curve, but they are much slower, or they might have the same response curve, but at much reduced maximum level. So this would be weaker induction, this would be slower induction. Okay. Again, it's schematic, it can be a combination of the two, but it can be also no induction at all. Right? And clearly we really wanted to measure precisely these parameters and see whether we see a bimodal distribution of response parameters for the normal cells and the persistent cells. And we bet on which of the two it's going to be. Some of us really like the fact that it's a scaling with time, and some of us thought that there will be no response at all, but at the end of the day, we were all wrong. So we went back to the microfluidic devices, and now we do this experiment. We have normal cells and persistent cells within the same population with this induction system, and we grow them, and then we add the signal. So we add the signal at the beginning of the experiment, and we follow the induction level, the response of normal cells in black and persistent cells in red, and they just fall on top of each other. So, and we can convince ourselves that this is a normal cell, and this is a persistent cell because here we added antibiotics, and this one died, and this one survived the whole course, right? But when we were looking at the induction curve at the beginning of the experiment, they just are indistinguishable from each other. So here you don't have a bimodal distribution, you have a unimodal distribution. Here, of course, you have a bimodal distribution. So it was not clear what was going on here. It was not clear because if this cell responds to the external signal, it should have died here. So it was not clear. But it looked as if, when we did this experiment many times, it looked as if there was a time scale, and here this is without antibiotic. There is a time scale where they are identical, but then after about an hour and a half, all the persist cells that are here in red just stop responding to the signal. So they are initially responding to the signal as normal cells, and here, in fact, is the decision taking not to respond or to become dormant. Right? So what it means is that if we dilute the cell, T equals 0 is defined by diluting the cell into fresh medium, giving them a growth signal. If we dilute the cell here and give them the induction signal only here, in principle, those cells are not supposed to be induced. So this is the next experiment that we did. Again, in the microfluidic devices, we just grow them, we give them a lot of nutrients, enable them to enter this dormant state, and then only after these the first 2 hours, only then we give them the induction signal, and now they are not going to respond at all. So the filters here are completely dormant. So this time actually defines when is a persistent switch occurring in single cells. And indeed, before they are not dormant, and afterwards they are dormant. So it means that when you dilute the cells and give them growth signals, even the ones that are going to be persistent, after a while, they are not yet persistent. So you might be able to get rid of them. And usually, typically, when you do an experiment with antibiotics, you dilute the cells from stationary phase, you wait a few generations at least, and then you put an antibiotic, right? So now, instead of waiting, as we always do, and we diluted the cell at T equals 0 immediately into antibiotics. Right? And this is the total population, and this is the persistence level. So when you dilute them immediately into antibiotics, you catch the persisters before they make their dormancy switch, and you kill many more of them than if you just wait an hour and a half, and then add the antibiotics after the dormancy switch has occurred. So, yeah, I think I'll summarize here. So, yes, actually we're having fun following these dormant cells, and we think that the understanding that you get from quantitative measurement like that can indeed help you find all kinds of ways in order to get rid of these persisters, and eventually find a way to generate viability of growth, which is a very efficient strategy both for phages and for antibiotics and for heat and God knows what. If you want to evolve such a strategy, then the substrate of the toxin antitoxin modules are ideally suited for that. So I'd like to thank all the people that did the work. You saw them in snapping at various stages of the talk, but this is the group. Mostly Eitan Rotem did the toxin antitoxin study. Irit Levin, together with Orit and Ofer, developed the scanner system, and Sivan did the phage work that I didn't talk about, but I thank all the collaborators, mostly Ofer Biam and Adiel Longer, his student, that did the stochastic simulations of the toxin antitoxin system, and Amos Oppenheim, who was a great collaborator on the phage system. Thank you.
Thank you, Nathalie. Question?
So this will be a role that will be in because of the accumulating error that
that's actually this example where if you have some small error in translation, then as long as the error still maintains the toxicity of the toxin, but decreases the binding depth, it might be an advantage. It might be an advantage.
But you didn't check.
We could, there are studies that increase this error rate. We could check whether it has an impact.
Any more? Okay, thank you again for the first.